And our third speaker will be Dr. Kaustev from the Department of Science and Technology of University of Nebraska-Lincoln. And so Dr. Kaustev is an associate professor, and he earned his PhD from the University of Albert, where he focused on food and cardiovascular health, followed by postdoc training at the University of Gulf. So he specialized in like food allergy and allergic protein characterization. So Dr. Kosdev is committed to understanding how dietary proteins and peptides influence human health. So I think without further introduction, I want to give the time to Dr. Kosdev, and so please move forward with the presentation.
Thank you, Dr. San. Am I audible?
Yeah.
Can you hear me, right?
Yeah, we can hear you.
Thank you for the introduction. So today, what I'm going to talk today, a little bit portion of what we are doing in the lab here at UNL. So my lab primarily works with food proteins, and when we look into the food proteins, when we, as previous speakers mentioned about different ways of looking at food proteins, but we looked how after cooking and processing, once the changes happens in the food proteins, and once you consume them, and once it's broken down into amino acid and peptide into the GI tract in the small intestine, what's their impact on human health, specifically looking on cardiovascular systems after absorption. So that's get absorbed in the small intestine and what's their effect on small cardiovascular health. We also looked into the physical properties of different proteins and how it manages the emulsion, foaming, agitation, all of those qualities for food applications needed, and how all of those affect the digestion and the bioaccessibility and bioavailability of peptides in the GI tract. On the other part of my research, we do work with the agronomists and try to see how plant domestication methods and soil nutrient management practices affects the protein expression in specifically plant proteins, of course, as we're looking into the soil nutrient management and how those affects our diet, specifically on the absorption of proteins and peptides. On the fourth approach, we looked into food fortification and bringing into skills of precision fermentation to make the protein-rich foods more affordable in case of that's to help its specific effect on human health. And also we're interested of long voyage foods, like if we go to Mars and someday what we're going to eat in this whole journey. So that's kind of things we start looking into as well. So today in the talk, I'm going to focus on these two topics, the theme 1 and theme 4, and try to give you a story specifically focusing on peptides. As I'm talking about peptides, we have this conversation with the previous speaker on bioactive peptides. So we look into bioactive peptides. That's what one of the major things in the lab we do. So these peptides, as you know, it's inert in the parent protein sequences, but once liberated through different ways of food processing or during gastrointestinal digestion, or could be naturally occurring from plant or animal sources. Once it can show biological activity. The two major challenges of these bioactive peptides are their stability in the GI tract. As they have proteolytic enzymes in the GI tract, they can't be broken down easily. Another point of that, all of those bioactive peptides, if not every one, the majority of them have bitter taste, so it doesn't really have an appealing application, not palatable for food applications. So we looked into a class of peptides. We called it isopeptides, or these are the isopeptides. So we can look into the normal peptide bonds is basically looking at the carboxylic group or amino group and conjugate together, form a normal peptide bond. But in isopeptides bond, it uses not the alpha carbon, but the gamma carbon of an amino acid, like glutamic acid, and make the peptide bond. The classic example is glutathione. As again, everybody familiar of the glutathione. We have this isopeptide bond between the gamma glutamate and the cysteine, and then the regular peptide bond between the cysteine and the glycine. So these peptides bond are formed due to this presence of this gamma carbon, and primarily it's glutamic acid that's present in the food. And these are called the dietary gamma-glutamyl peptides. Now, if you look into that, these γ-glutamyl peptides, they are abundant in beans, cheese, onions, and soy sauce, a lot of also in those fermented foods. And they produce a sensation called kukumi sensation. So people often confuse between umami and kukumi. So that's what I put it up there. So umami is you give a deliciousness or pleasant flavor taste, where kukumi is a taste enhancer. It's a long-lasting effect of a specific taste. So these these peptides, they actually elicit this kukumi taste through activation of calcium sensing receptors that's present in the posterior end of your tongue. And due to this gamma bond, they generate resistance to the GI digestion. So we start looking into these peptides. And when we start looking into these peptides, we also see these kukumi substances that activates this KSR, the calcium sensing receptor, that have neural stimulation molecules in type 3 cells. So it's pretty much obvious that it's activates through this one particular pathway when event consumed. And when we look into the KSR a little bit closer, we found out that this particular receptor, the calcium sensing receptors, have a calcium binding domains, but then also there are other domains where these peptides are mostly binding, and few other research has been shown that these peptides can allosterically activate this particular receptor. Now, KSR is present all over the mammalian body, and that's what start asking the question, if KSR is present all over the mammalian body, not only in tongues, in the GI tract, in the lungs, and what it's doing. So we start looking into this activation of KSR through gamma EV, and we found out that it can actually inhibit in inflammation through the activation of KSR in the GI cells. Now, as I mentioned before, my lab starts looking into vascular inflammation and specifically atherosclerosis. And when we start looking into atherosclerosis, we know that the initial vascular inflammation have a significant role in the progression of the disease atherosclerosis. And we thought that this, with activating the same pathway, that the gamma-glutamyl peptides reduces vascular inflammation and can develop anti-atherosclerotic effect. So we use these peptides called gamma-glutamyl valine, gamma EV, as our model peptides to study this one. So we looked into the vascular endothelium. So endothelium that's the innermost layer of the vasculature. And we start finding if the KSR is present there or not. So because KSR is our target molecule. And we found out that KSR actually presents in the endothelial cells. High portion is also in the membranal fraction, but there are also a portion in the cytosolic fraction. So this confirmation of that KSR presence in the endothelial cells gives us the confidence to go forward and see what's their role in vascular inflammation. So we use the hematopoietic endothelial cells, and we treat these gamma EV in 3 different concentrations for 2 hours. And then we give it a TNF-alpha treatment to induce inflammation, and looked into different cell adhesion molecules and the chemokines and cytokines. So going forward here, you can see that this is the negative control. We did not add the TNF here. This is the positive control. We have the TNF, achieving increase in inflammatory biomarkers like IgM1. And this is the response with the different doses of gamma EV. And we can see there is a reduction trend, but however, it's not statistically significant for IgM1. But for other 2 biomarkers, VCAM1 and E-selectin, we see a substantial decrease in 1 millimolar concentration of gamma EV. We looked into the chemokine MCP1, and we see a similar reduction, even with a 0.1 millimolar concentration of gamma EV, as well as for IL-8 and IL-16. So these particular peptides is showing anti-inflammatory effect. But what is KSR responsible for that? To ask that question, we use a KSR-specific antagonist, MPS 2143, and then we hypothesize that this KSR antagonist will take out the KSR, will block the KSR access for gamma EV. As you can remember from VCAM1, we see a reduction in inflammation when you have the gamma EV. Now you add this MPS 2143 for VCAM1, E-selectin, and MCP1, And we see that effect coming back. So there is the anti-inflammatory effect of KSR is nullified when you add this of gamma EV is nullified when you add this NPS 2143. So basically, the gamma EV is acting through that KSR receptor. So now, to go to the bloodstream, we need to see if the KSR actually reaching the bloodstream. So we see the bioavailability studies of gamma EV through CACO2 cells that's grown over 21 days. And we use this same dose, that 1 millimolar dose, that's the most effective dose that we have in the previous study, and use for 1 hour and 2 hours of incubation. We take this media down here in the basal layer, and then did the mass spec to identify if it's transporting or not. And it does transport through the CACO2 cell monolayer, and after 2 hours, we get a PEP value of apparent permeability value of 1.56 without damaging the intestinal cells. As you see, there's no change in the 0-1 expression, the tight junction proteins. So all of these part of the study can summarize that we see that gamma EV, that's Kukume enhancing peptides, can reduce vascular inflammation through the activation, through the reduction of different biomarkers, as well as reduction in chemokine and cytokines. And we also see this effect is KSR mediated, and these peptides can actually cross the epithelial, gastrointestinal epithelial cells and can go to bloodstream. We also look these things in an animal study. So the animal bioavailability, once we give a gavage of 100 milligram per kg body of gamma EV, we see after 10 minutes to 12 minutes, we see an increase of 5.26 micromole in the jugular vein. So it's actually going through the circulation. So that gives us the confidence to move for the animal study. So for the next phase of animal study, we looked into the ApoE knockout mice, so apolipoprotein E knockout mice, which cannot process the lipids in their system and then produce atherosclerosis in the arteries. And we see what we want to see how the effect of this gamma EV affects on the plaque accumulation. When looking to these studies, we break it down into 4 different groups. We have these low-fat diet groups, and then a high-fat diet group, and then high-fat diet with 2 different doses with 50 and 150 milligrams of gamma EV. So it's roughly a 20-week-long study. We start with the pre-treatment of gamma EV, and then continue with the high-fat diet. So this is what the results looks like in the aortic arch and area. So this is the aortic arch area. You can see that's a low-fat diet. You do not see a lipid accumulation. However, in the high-fat diet, you see a lipid accumulation that's happening, which is reduced with the low-fat diet and low dose of gamma EV as well as a high dose of gamma EV. When we quantify it, you see a significant reduction on the lipid accumulation in the aortic arch after the gamma EV treatment. We see a significant reduction on the same biomarkers, VCAM1, ICAM1, as well as on LOX1. Now, remember, ICAM1 in the cell study, we did not see a reduction in ICAM1. However, in the animal study, we see reduction in both ICAM1 and VCAM1, and also in LOX1, which can be only read in an animal study. Once we do that, we move to these cytokines and chemokines. We see a significant reduction in MCP1. We also see the same thing in the cell study. We also see a reduction in RANTES, but that happens when you have atherosclerosis. But with the treatment of gamma EV, we did not see it's coming back. We see a reduction in TNF-alpha with the gamma EV treatment, and to our surprise, we see an increase in IL-10 expression. Now, IL-10 have an anti-inflammatory effect. We see an increase in IL-10 expression. So looking into all of these things, we can tell that these peptides have an anti-inflammatory effect and can reduce lipid accumulation. And this anti-inflammatory effect may be modulating through KSR. We are doing additional study to find out, is it really KSR mediated, or some other things are involved as well. But now when we start looking into if it's the KSR is the only sole responsible activation is sole responsible way of doing this anti-inflammatory effect, we found out that's quite a bit portion of these gamma EV is actually going to colon, like roughly around 75% that's going to the bloodstream, and remaining 24% that's actually going to colon. Now, the question is that what's this this gamma EV that is reaching the colon? What is their effect, and how, as they're reaching intact, how it's interacting with the gastrointestinal microbiota? So we start looking into the beta diversity. Again, thanks to the previous reviewers talks about what is beta diversity already, so I saved my time there. So when you looked into the beta diversity, we looked into the 4 different indexes with Bea-Curtis, J-Curt, weighted unifrac, and unweighted unifrac. And if you focus on these 2, you can see this is the low-fat diet, the purple group up here. You see the blue one with the high-fat diet, and there is a shift in the microbiota composition in high-fat, high-dose group, as well as there is a little bit change in high-fat, low-dose group as well. But that shows that with this treatment, we are seeing a significant change in the gut microbiota populations. And when we look into the abundance of the different gut microbiota, and what 2 specific things that bloom out in our study is that with the high-fat, high-dose, we see a specific pattern of increasing Akkermansia as well as Dubicella. Now, this Akkermansia and Dubicella is really getting high up in the high dose diet of gamma EV, and we see other specific bugs that's actually going up or having some changes with the treatment of gamma EV, but specifically Akkermansia is what gets our interest, and we take the cecal continent PCR for Akkermansia muciniphilia to confirm our results. We see that the similar effect, that there is an increase in Akkermansia muciniphilia in high-fat, high-dose group once treated with the gamma EV. Then, if we go back to the literature, there are evidence that Akkermansia give a protective effects against atherosclerosis, and it's primarily, a lot of research has been done on the lipid lowering effects on Akkermansia, and sometimes this polyphenol-rich extract that's increased Akkermansia have an impact on diet-induced obesity models. So this particular bug is have a beneficial effect in atherosclerosis setup. Now, how the mechanism behind it, we really don't know yet. Now, we did a correlation analysis between all of our biomarkers that we looked into it, and with Akkermansia, and we can see that Akkermansia muciniphilia was negatively correlated with ICAM1, VANTIS, as well as the aortic lipid deposition, and positively correlated with HDL and IL-10. So that gives us the direction that there are some effect that's happening after the gamma EV treatment, which is gut microbiota dependent, and there will be some effect that is probably gut microbiota independent. So once it absorbed in the small intestine through the KSR, potentially, again, we still trying to find out if it's KSR the sole responsibility of other receptors as well, that's happening, and that's having an effect in antimicrobial activity. And the portion which is not getting absorbed going to the colon and through Akkermansia, increased Akkermansia abundance, potentially affecting this anti-inflammatory effect. Now, we wanted to see if it's really having a specific effect, the gamma peptide specific effect in Akkermansia abundance. So we took it to the in vitro system. We supplemented Akkermansia muciniphilia with gamma EV and look at their growth, and you can see with 10 millimolar supplementation of gamma EV, we can see a significant increase in the growth of Akkermansia with the gamma EV supplement. Now, 10 millimolar dose seems a bit higher, but if you go back to the calculation, how much we are actually retaining in the colon in a single day of treatment, if it's work goes for 20 weeks, and 10 millimolar can be achieved pretty easily. So the dose is pretty much achievable in an in vivo setup. So it's a physiologically relevant dose, we feel. So this study is concluded so far that gamma EV treatment selectively reach Akkermansia muciniphilia in high-fat feed apoe mice, and it's negatively correlated with ICAM expression and aortic lipid deposition, which continuing the story of lipid metabolism with the acromegalia, increase in acromegalia, as well as these can be the increase in the growth of acromegalia and mesothelioma in vitro suggests that acromegalia is utilizing gamma EV for its growth. Now, connecting that back to the food story, when we start looking into different kind of food sources where we can find gamma EV or other gamma peptides, we found Great Northern beans as one of the richest source of these peptides gamma EV. I say richest source, but it's pretty small still. It's only 52 nanogram per milligram of bean proteins. So the major challenge, if you wanted to have this as a food item, is that there's very low abundance, there's very low amount in there. So you need to probably eat kgs of beans to get that amount of that we are looking at. So that's where we go into precision fermentation or different other food processing ways, how we can increase the abundance of these gamma peptides. So we looked into one specific bacteria called Limosobacterobacillus leuteri, LTH5448. So this specific bacteria has a gene called glutamyl cysteine ligase, or GCL. So this glutamyl cysteine ligase for the specific bacteria, they actually ligates, so not proteolysis, it's a ligation reaction, ligates the glutamyl peptides, glutamic acid with the conjugate amino acids, and produce glutamyl peptides. And we see an increase in peptide concentration of this specific peptide concentrations once we do a fermentation for up to 72 hours. So we take dry beans and this specific bugs and did a liquid fermentation as well as solid state fermentation. The results you see with right now is through a solid state fermentation. So once we see the increase of the peptide concentration, we looked into the different peptides, gamma glutamate peptides is produced after fermentation. We see all of these peptides like gamma EC, EG, EY, EL, EF that is producing. The gamma EV also we detected, however, we did not see a significant increase, but we also detected all of other gamma peptides. So it's not one specific gamma peptides we're looking at, we are a pool of gamma glutamyl peptides that we're looking at. So we increase the pool of gamma glutamyl peptides, but can we retain the biological activity? Then we start looking into going back to the same cell model and induce inflammation through TNF-alpha. I looked into the VCAM1 expression. We see both 42 hours and 72 hours fermented samples decrease the expression of VCAM1 as well as MCP1. So what we see with the pure peptides, we can see the same one when we produce the fermented product. And when we taste that in the same cellular system, we see the similar effect. And however, the gamma EV was low, so we start looking into different ways of increasing. Now we finally found ways how to pre-treat our samples that can increase gamma peptides, specifically for gamma EV, now roughly around 19-fold from our solid state fermentation. And these are the liquid state fermentation. Now with these fermented products, you can probably use it as an ingredient to make different kind of food items. So we are presently making cookies out of it, which is enriched with gamma peptides. And we did some initial sensory study, which shows that the cucuminous of this whole product is also increased. So that gives us that idea that we can probably use a fermentation away to increase this specific peptides. So now this study so far concluded that we increase the abundance of gamma glutamate peptides through this precision fermentation method. And these peptides that produced through this fermentation actually exhibit the health beneficial biological effects, the anti-inflammatory effect on vascular cells as we see with the single peptides. So that's what I want to stop here. This is the students who do all the work. So I really believe in diversity in the lab. So we have different peoples from different continents, and that gives us ideas how to work coherently, as well as my collaborators and the funders for this particular research. Thank you very much for your time. I'd love to take any questions that you may have.
Very good presentation. Thank you, Dr. Carl, Steph. And so, yeah, um, question time.
Dr. Magenda. Um, very
Right.
nice talk, and I'm just curious to say, so how widely? I mean, I say like so. I think gamma, peptides. For example, like gamma EV, these dipeptides. From your presentation, I think it's mostly existed in fermented food, right? So
Mhm.
is it also like maybe existed in some other kinds of protein-rich foods, except like fermented food?
We screened a lot of foods so far. However, their abundance or their amount in the non-fermented food, let's put it that way, their abundance is pretty low. They are easily found in the fermented food. So I think that it's more of a question of abundance. You see a lot of these peptides in the fermented foods. Recently, a paper came out from Kyoto University, and they identified these peptides also in rice wine vinegars. So we see a lot of these peptides in mostly in the fermented food. But yes, you are right. There are like our initial study in the beans, we see also in onions, we see a little bit of these peptides as well. But they're abundant, as I said, it's pretty low.
Yeah, I think, from your talk, I think you found these peptides actually has a relatively higher amount in Northern beans that
Mhm,
was grown in not in Nebraska, Lincoln, I think.
mhm.
Okay, yeah. I mean, that's like, provides a new angle actually when we develop this from, beans based, type of plant-based food from for in terms of the health benefits. Thank you.
Yeah.
Yeah. Yeah. Any more questions?
I can add one more. Maybe I missed in your talk. What about the half-life of these peptides? Like, once it's
Uh,
absorbed into the blood circulation.
we looked into it's roughly around 90 minutes.
Wow, it's pretty good actually. I
Yeah.
mean, many peptides you could list only a few minutes.
Yeah, so we see the peak around 20-minute time point, like sorry, 15 minutes time point, and then it's kind of phasing it out, but we can detect up till 90 minutes. Now we looked at that 90 minutes. I'm talking from the. I think that data, if I remember correctly, that's coming out of not in the jugular vein. I think jugular vein we did not see after 60 minutes or an hour or so, but in the portal vein we detect up to 90 minutes.
Okay.
Um, okay.